This is a lecture concerning one of the chapters in class 10 of the Karnataka State Education Board. It is loosely based on the NCERT textbook. As you know, Jawaharlal Nehru Planetarium is not only involved in education in astronomy and popularization of astronomy, but it is also broadly involved in disseminating science at all levels for both student community as well as the teaching community. So towards this end, there are several educational programs that Jawaharlal Nehru Planetarium, Bengaluru conducts all through the year, and we have developed several modules related to high school science and mathematics, college science and mathematics, and so on. Now this particular lecture is to facilitate students and teachers to teach one particular chapter on acids, which is part of class 10 syllabus. So let us make one point clear at this stage. This is not a substitute to the classroom teaching. This is not a tutorial session either. This is basically a session about acids, which is aimed at conveying the exciting things about acids through some experiments and demonstrations. This particular lecture supplements the science teaching classes that are formally conducted in your own schools. So let us get started with the first aspect of acids. So where do we begin? So we begin with things that are very, very familiar to us. As you can see, this is tamarind, this is lemon, and this is tomato. And who would not have tasted as young children? I mean, even as adults, we enjoy tasting this. It's a paste made out of tamarind, salt, sugar, jaggery, depends on your taste. It's a mouth-watering item that we all have enjoyed as children. Now, what is common to all these? Now, one thing that is common to all of these is that they all have sour taste. The sour taste basically comes from the fact that each of these items, tamarind, tomato, lemon, they contain an acid, or it is more than one acid. In fact, the root meaning of the word acid itself is sour to taste. It doesn't mean much. So during the course of this lecture demonstration, we will see what exactly we mean by acids. Now, acids form a very, very important group of compounds, not only in chemistry but also in biology. In fact, without some of these acids, it's impossible to think about life as we know. So we have amino acids, then we have fatty acids, we have acids in the form of certain vitamins. Vitamin C is ascorbic acid, vitamin A is retinoic acid. Then we have the hereditary material, DNA and RNA, which are deoxyribonucleic acid and ribonucleic acid respectively. So all these host of acids, they form part of our own cells, and thousands of reactions that take place within our body which keeps us living all the time is mainly because of the presence of these acids. Now, you take for example the amino acids. There are 20 amino acids known to us in nature. Out of these 20, 11 amino acids are produced within the human body. There are 9 other amino acids which are extremely important, which we ingest from the food that we take. And these amino acids are rightly called the building blocks of proteins, because without these amino acids, proteins cannot be made, and proteins have specific biological functions within the body. It could be making the skin elastic, or it could be making the muscles springy, and so on. And these characters come about because of the proteins which are actually produced with the help of amino acids. So the role of these acids cannot be further emphasized. And when it comes to some of the behavioral aspects in the animal world, especially insects, we see that acids, which are basically corrosive, they cause a little bit of burn when they fall on the flesh or skin, and sometimes they form inflammation. They can be very, very unpleasant. Now, these acids are produced in the insect world as a method of self-defense. So, there are several species of ants which actually inject formic acid when they bite, and because of that we feel the unpleasantness, and sometimes a small niggling pain which is because of the formic acid, and our body is reacting to the formic acid which has been injected by the ant. And there are certain species of ants which have specialized organs to squirt acid at the predators. Whenever they feel that they are threatened by a predator, they actually squirt acid on them and they escape while the predator struggles to overcome the effects of the acid. So, in a very large scenario, as far as biology and life is concerned, acids play a very, very important role. So, this is one of the reasons why we need to learn about acids. Now, to start with, we have acids which are made within the bodies of living systems. For a long time, people thought that these acids need organs to be made. Therefore, making these acids outside the realm of living systems was unthinkable. But today, our own understanding of acids is so vast, and we have come to realize the importance of acids so much that of all the chemicals that are manufactured, they are synthesized in industries, acids form the bulk. In fact, hydrochloric acid, nitric acid and sulfuric acid are three of the major acids which are produced by countries all over the world, and in turn these acids form as raw materials to produce thousands and thousands of other products which are useful to us in our day-to-day life. So much so that sulfuric acid is often called the king of chemicals, because that forms the basic raw material to prepare thousands of other very useful compounds to us in our day-to-day life. Now, to begin with, as I mentioned, the word acid itself comes from the taste, that they are all sour to taste. So, some of these acids, like the acid that is present in the lemon, which is citric acid, or the acid which is present in tamarind, which is tartaric acid, these are comparatively harmless. So, when you taste, your tongue does not undergo any kind of damage. But there are acids like sulfuric acid or hydrochloric acid which are extremely corrosive, they are very dangerous, and they can cause burn in the flesh, and that can damage organs permanently. Similarly, we also have acids which are not sour to taste. So, in the beginning, all acids that we came across were sour to taste, and as we learnt more and more about acids, we found that not all acids are sour to taste. In fact, among these 20 amino acids that I mentioned to you, there are several amino acids which taste sweetish, some which are saltish, some which are bitter, and there are some acids like retinoic acid whose taste cannot be deciphered at all. Different people have different sensation of taste when they are asked to taste retinoic acid. So, this subjective way of classifying a compound as an acid on the basis of taste is not a very scientific one, and also it is not very, very safe either, because there can be acids that when you try to taste, they can harm your tongue. So, this subjective sensation of taste is not a good way to classify acids. Then what is the scientific way? What is the safe way, or what is an objective way of classifying a compound as an acid? So, generally we look for a chemical reaction in which all acids react with a particular compound in a very, very specific manner, and this specific chemical behavior of the acid will reveal that a particular compound is really an acid. Therefore, we need on the one hand acid, on the other hand another chemical which specifically reacts only with acids. So, this particular chemical that we add to the acids should not react with any other known compound to give the same observable result that we see in the case of acids. Therefore, we look for a very specific chemical reaction between an acid and some other compound in order to find out whether a given chemical is an acid. And one of the oldest way of finding whether a compound is an acid or not on the basis of this specific chemical reaction is what is known as the blue litmus paper. So, this is a litmus paper. It is paper that is coated with a compound. It is actually a mixture of compounds. There are about 15 different compounds that make up this blue litmus, and these compounds are actually dyes, meaning that they impart color, and they are extracted from lichens. So, lichens grow on the bark of trees, as you would have observed. One particular species of lichens known as Roxella montagni is used to extract these 15 colored dyes, and a mixture of these dyes gives what is known as a blue litmus. Now, this blue litmus reacts in a very specific way with acids. So, like I mentioned to you earlier, there must be a specific reaction between all acids and this blue litmus, which gives us the same observable result. So, let us see what the observable result is. So, first I take here the test tube a small quantity of vinegar. You know, vinegar is one of the commonly used kitchen ingredients. It is chemically called acetic acid. So, I add acetic acid to this test tube. And now we have this blue litmus paper, which I shall dip in the acetic acid. Observe that the color of the acetic acid is colorless. It looks more or less like water. This is blue litmus. So, I add blue litmus into this, and you observe that the portion of the blue litmus which is dipped in acetic acid has turned red, whereas there is no change in the color of acetic acid. Now, alternately, I can also use a blue litmus solution instead of the blue litmus paper. So, here I have blue litmus solution. Now, the purpose behind this particular demonstration is the beauty that goes with the color change. So, the moment we add a few drops of this, you will find that the entire acid becomes reddish. So, this is a specific reaction. This is what I was talking to you about, specificity of chemical reaction between acids and litmus. But we have so far seen only one acid. So, let us see if we get a similar result with other acids that are known to us. So, here I take citric acid. So, citric acid is obtained from lemon juice. Lemon juice is a mixture of many things. Citric acid is also one of them. I should just dilute this by adding a little bit of distilled water. Not that it is essential, it is just to make the quantity of the acid more, so that I can dip this blue litmus paper into it very easily. By the way, when you handle these chemicals, you should be very careful if you are doing this experiment yourself. If your hand is contaminated with acid, then the litmus solution on the litmus paper will react to that, and it will be rendered useless later on. So, there we go. So, that is the blue litmus which has been dipped in citric acid. We find that the blue litmus, the portion of the blue litmus paper which was dipped in citric acid has turned red, just as in the previous case when we dipped blue litmus into what acid was that? The acetic acid. So, whether it is acetic acid or citric acid, the reaction is the same. So, again, like we did before, I will have a few drops of this blue litmus solution into this, and you will find that citric acid again turns blue litmus red. Therefore, whatever the nature of the acid, chemically citric acid is very different from vinegar, that is acetic acid, but their reaction with blue litmus is exactly the same. So, this is a specificity in the chemical behavior that we look for in order to classify some compound as an acid. So, any compound which turns blue litmus red has to be an acid. So, just to make things better, just to make things more general, like they say, 3 times is a good time to stop. So, let me do this a third time in another acid. Maybe I'll try this with hydrochloric acid. So, I have hydrochloric acid there, and add a few drops of hydrochloric acid. Okay. And let me drop this blue litmus paper into this acid. So we will find that it turns red. Right? Therefore, we can conclude from these 3 experiments that acids can change blue litmus to red. So this is one way of identifying an acid. So now we don't have to anymore depend upon my ability to taste the sour taste of acid. Let us summarize now. So what we see from these set of experiments is that whether it is hydrochloric acid which is industrially produced, or vinegar which is also industrially produced, or whether it is citric acid that is obtained from a plant, or it can one can even try with tartaric acid. As we mentioned about this, but we never used it. So let me just use this tartaric acid, and again when we have this blue litmus paper dropped into it, you'll find that it turns red again. Okay, blue litmus turns red. So whether the acid is of plant origin or animal origin, or it is synthesized in an industry or a laboratory, the acid character is always common. The common character is that they all react with blue litmus and changes the color of blue litmus to red. So this is one class of reactions. These are called indicator reactions because the reaction indicates whether or not something is an acid, and the indicator in this case we have used in this case is blue litmus. So there are other kinds of indicator reactions. We will look at these next. So as we saw now, blue litmus reacts with acids irrespective of the origin of the acid, irrespective of the chemical composition of the acid, in exactly the same fashion. It turns its color from blue to red. So this is a definitive test for an acid. So not only a definitive test, very often even in a very literary sense or a poetic sense, we say that something is a litmus test. When we say something is a litmus test, it means a definitive test. So blue litmus turning red is one of the definitive tests of an acid. Now likewise, we have a few other definitive tests for acid. These are also indicator based reactions. Just as blue litmus is an indicator, there are other kinds of indicators, and these indicators can again be human made, they are synthetic, made in the laboratory, or they can be produced by natural resources. One of the commonly used natural resource based indicator is the extract from this red cabbage juice, red cabbage. So all you have to do is to extract red cabbage juice from this leafy part, crush it using a mortar and pestle, and then dilute it with distilled water, and you have and filter it out. So what you have basically is some kind of a solution which is purple colored like this, and this is red cabbage juice. Now red cabbage juice is again a mixture of many compounds. It's very difficult. I mean, to my knowledge, I don't know of any example in nature where you get pure compounds. most of the times you have a number of compounds which are mixed together. So whether it is a part of a leaf or part of the flower or part of a root, it's a complex mix of different compounds. Similarly, the red cabbage juice is also a mixture of different compounds, but out of this mixture there will be some specific compound which reacts with all acids in a very specific manner, giving rise to an observable result which is common to all acids. So let us see what this observable result is. So we take once again some acid, and what do we take? So let us start with citric acid, the lemon juice. Now the reason why I am picking up vinegar and citric acid for many of the demonstrations is that these acids are very easy to handle, and since we are only demonstrating the chemical properties of acids that are common, it does not matter what acid we take, and since these are very safe, you can also repeat these experiments at your leisure in your classroom. So I have red cabbage juice in this boiling tube. Let me take a small bit of this red cabbage juice and add it to the acid here. So this is the color of red cabbage juice before we add to the acid, and you see that the acid, the color of the acid or the color of the indicator red cabbage juice changes. So it has a pinkish appearance. This is a kind of a definitive test for an acid. So red cabbage juice undergoes this kind of a color change. Now we can also repeat this experiment with another acid. So no marks for guessing what I am going to take next, because it is going to be vinegar. As I told you, it is very safe to handle, therefore I am using vinegar, and to this we add a small quantity of the red cabbage juice and observe the change in color. So once again it changes its color to pink. So this color change is very specific to the reaction between some compound which is present in the red cabbage juice and the acid. So you can use naturally available indicators like this. It is real fun for you to carry out these experiments. In fact, this blackberry juice, blackberry is ನೇರಳೆ ಹಣ್ಣು in Kannada. So this blackberry juice also gives you some very interesting changes in color. Let me show you with one of the acids. So let me take for a change hydrochloric acid, which is here. I take a small quantity of hydrochloric acid into this test tube. Maybe add a little more liberally so that we can see the color change very easily. So I am taking from this blackberry juice a small quantity of this. In all these indicator experiments, you actually don't need a large quantity of the indicator. Just a couple of drops would be more than enough to observe the changes in colors. Now we have the colorless hydrochloric acid there, and to this we add blackberry juice. See that again it changes the color distinctly, and that is an indication that the liquid inside the test tube is an acid. So you can have lots of fun doing similar experiments. So I suggest that you try with this blackberry juice, red cabbage juice, and then you have beetroot, and again it will be in the form of beetroot extract, and also the black grape juice. Now you can try this with various kinds of fruits and vegetable extracts, make a solution out of that, and play around with acids that are available at home, especially those acids which are available in the kitchen are obviously very safe. So we have 3 of them, tartaric acid from tamarind, hunse hannu, or we take citric acid from nimbe hannu, which is lemon, or we can take vinegar, which is also available off the shelf in any departmental store. So, these 3 acids will form the samples on which you can try out these experiments with natural indicators. So, to sum up this part again, I would like to impress upon you that indicator based reactions are essential to identify whether some liquid is an acid or not, and these indicators are a safe way to do that, better than tasting for sourness. And secondly, many of the naturally occurring materials like fruits and vegetables, they contain compounds that react with acids in a very specific manner, therefore they are very good indicators. So, if you know that something is an acid, then you can find out whether some particular vegetable or a fruit or a flower has indicators which react in a specific manner. Or if you want to try the other way, if some extract from a flower, fruit or root or any other part of the plant is contains compounds which are like indicators, then you can use these acids to test against them. So, this acid and indicator based reactions help us to identify one against the other. Now, I end this particular discussion on indicator based reactions which will help us to recognize whether something is an acid or not with an experiment with methyl orange. I am performing this experiment only because this is also mentioned in the textbook, therefore you might be curious to see what the methyl orange reaction would be like. So, we take methyl orange in this test tube. So, we would like you to observe the color of methyl orange before adding the acid, and now we add a small quantity of acetic acid to this, which is vinegar. So, the moment we add acetic acid, it again turns pinkish. So, this is again an indicator based reaction between methyl orange and acetic acid, and just to make sure that every acid will react with methyl orange exactly the same way. So, let me take another acid, which is citric acid here, small amount, and then I shall dilute it with distilled water. Distilled water should always be used for diluting acids because of some reason which I will discuss a little later. So, for the time being, assume that distilled water is the one to be used. Tap water should not be used for diluting acids. So, there we add a couple of drops of indicator, and you see that the acid changes its color once again. So, this is again an indicator based reaction. Now, if all acids were to react with indicators in a specific manner, the question we ask is, would there be any difference from one acid to another acid? The answer is yes. It is just like studying the human behavior. At one level, all of us are human beings, so we share certain behaviors that are common, but at the individual level, we find that everyone's behavior is very different from somebody else's. In the same way, while we have citric acid, nitric acid, hydrochloric acid, tartaric acid and so on, all of them behave with blue litmus in the same fashion. There are certain reactions in which each acid behaves separately. So, just as knowing whether something is an acid or not is important, it is also equally important to know what kind of an acid a particular compound is. So, for that we need to study some more chemical properties of acids, which will help us to, at one level, generalize all the properties of acids, and at another level it will also help us to differentiate one acid from another acid. So, this is what we are going to do in the next section. Now, one of the chemical reactions which we take to begin with is the following. So, let us take the reaction of acids with metals. Now, one of the metals that I will be taking to start with is this magnesium. So, magnesium is generally obtained as magnesium ribbon. It is very brittle and also highly malleable. As you can see, it can be stretched into sheets and it can be very easily broken. Now, what we will do is the following. We will take this sulfuric acid, hydrochloric acid. Yes, I have hydrochloric acid here, and we add hydrochloric acid to this test tube. A small quantity is sufficient. When you do experiments with free metals like this, very often the surface of the metal would have reacted with oxygen present in the air and formed a coating of oxide. So, they are not very good for chemical reactions. And secondly, even if the reaction takes place, the reaction could be because of the metal oxide which is on the surface rather than the metal itself. So, what you do, this is a good chemistry lab practice. So, you take sandpaper or the emery paper and you scrape the surface of the metal before you add this to acid. So, I am going to scrape this a little bit, and once we scrape, we find that the metal surface becomes shiny. So, as you can see, the metal surface becomes shiny. Now, I add this to hydrochloric acid. Now, observe closely what happens to this. So, you see the hissing noise, you hear the hissing noise and you see lot of effervescence there. There is a vigorous reaction taking place between magnesium and hydrochloric acid, and in the process it is also liberating some gas, as you can see there. Okay. Now, this is typical of all acids. Magnesium reacts with acid and it forms these bubble like formation and also releases some gas. Now, let me repeat this experiment with another acid that we have. This is an exothermic reaction. So, when I touch the test tube, it had become very, very hot. So, these reactions are also extremely exothermic, which means they liberate lot of heat in the process. So, let me take another acid. This time what do I take? I take acetic acid here. No, yeah, acetic acid. Remember last time acetic acid and hydrochloric acid, both of them answered the blue litmus test in exactly the same way, showing that as far as their acid character is concerned, there is no difference between them. So, now we are going to add a small bit of magnesium ribbon to this. So, as before, I take the magnesium ribbon and then scrape the surface so that all the oxide coating is gone. And put this into acetic acid. So, you observe that magnesium is again reacting with acetic acid. It is forming those bubbles that we saw with hydrochloric acid, but for comparison you will find that the effervescence that we see in hydrochloric acid is a lot more than what we are seeing now in acetic acid. So both are acids, both are showing a common chemical reaction with magnesium, but the rate at which the reaction is taking place, it is different. So in one sense the two acids behave exactly identical, that is they react with magnesium in the same fashion, but at the same time the difference is that one reacts vigorously and the other one does not. Just like human beings, there are differences in the way we react, right? So in the same way the acids also show this differences in their behavior. So there are certain acids in which this magnesium may not react at all. So let us try with this citric acid, and so we have the citric acid, and I drop a little bit of magnesium. So once again I scrape the surface, and add this to citric acid. So again in citric acid you find there is a reaction, magnesium is reacting with citric acid and forming bubbles on its surface, as you can see there, the bubbles are rising up, but this is less vigorous than what vinegar showed. So even though we have vinegar and citric acid both reacting with magnesium, but the way in which they are reacting, the rate at which they are reacting, they are different. So this is one way to differentiate one acid from another acid. How quickly an acid reacts with a particular metal could be a way to find out the differences between acids. So what is happening here is the following. I will just write down the equation with respect to hydrochloric acid, which is much easier to follow, plus Mg gives rise to MgCl2 plus hydrogen gas. So the magnesium displaces hydrogen from hydrochloric acid and forms magnesium chloride, which is a salt, plus hydrogen gas is liberated in the process. So the bubbles that you saw rising up in this, those bubbles are the bubbles of hydrogen gas. This is a typical reaction between metals and acids. Almost all acids show this reaction, but the difference is that some react immediately and very vigorously, some react very slowly and less vigorously, and there may be some which react only when some external energy is supplied in the form of heat. But all the same, when metals react with acids, this is a reaction that you see, this is what we call the displacement reaction. Now let us move over to another reaction. Perhaps this reaction in this entire discussion about acids is the most important one as far as I am concerned. So let us try to do this experiment first, and then we will discuss the observations that we make. So far we have seen two common properties of acids, one with the blue litmus indicator and the other with the metals. So in the case of metals, we have seen that metals react with acids to liberate hydrogen gas, and in the process metals replace hydrogen, forming a metal salt. So in a similar fashion, there are other common properties of acids. and one which is very important for us is this particular one, the electrical conductivity. We have let us do an experiment, a series of experiments in fact, and in these experiments we will see what is it that we observe, and at the end let us try to understand why these observations have been made, which will tell us why acid behaves in a particular fashion. So, let me explain the electric circuit first. So, we have two carbon electrodes here, and these two carbon electrodes are connected through a power source, a 6 volt DC supply, and in turn that is the circuit is completed through a bulb. So, if everything works well and if the circuit is completed, the bulb should glow. So, now with nothing in this beaker, there is nothing in this beaker. So, we have these two electrodes and I switch on the power supply. Observe that the bulb does not glow. Now, you may say that this is very obvious because there is nothing in the beaker and the electric circuit is not completed. So, let me now take a piece of wire and connect the two electrodes directly. So, when I connect the two electrodes directly, you will observe that the bulb glows. Okay? Observe that the bulb glows. So, even though we have these two electrodes with a certain air gap, the current still flows and the circuit is completed because we are shorting this through this piece of wire. It is as though these 2 points have been connected directly by this piece of wire there. So, now let us put something into this beaker. I have distilled water here. Like I told you earlier, distilled water is very important for many chemistry experiments because distilled water will not affect some of the results that we look for. So, we have distilled water there, and maybe I will take out a little bit of this distilled water. It is a bit too much, and I switch on the supply. There is no the bulb still is not glowing. Just to make sure that I have switched on the power supply and you trust my words, let me once again short these two, connect with this piece of wire, and you will find that the bulb glows. So, what this means is, if I replace air in the beaker between the two electrodes, if I fill that with the distilled water, it is not making any difference to our experimental observation. So, air as well as distilled water, both of them behave in a similar fashion as far as completing the electric circuit is concerned, and we say that distilled water or air they are not good conductors of electricity. So, now acid comes into picture, small amount at a time. You know, when acid is added to water, one should add small quantity at a time, and that should be followed by constant stirring. So, I stir this and then add more of this. Now, you can actually see that when I stir, the bulb is just about glowing. Can you see that? The bulb is just about glowing. So, what this means is that adding acid to this distilled water brings about the same effect as using a piece of wire to connect the two electrodes. It is as though the two electrodes which are physically separated are somehow now connected with some kind of a wire, except that the wire is not in the form of a wire that we know, but that wire is made out of the acid that I am pouring into this. So, I pour more and more acid into this, and you will observe that the bulb becomes slightly brighter. So, as I stir, you see that the brightness keeps changing. So, now it is pretty bright. Okay. So, to summarize what we have seen so far, we see that when there is nothing in the beaker except air, the bulb does not glow, and it is very obvious that the air is a bad conductor of electricity, as you have already learnt. Therefore, the circuit is not completed. But when you add distilled water, even though there is a medium connecting the two electrodes, which is more visible than the air, we find that the electricity does not still completely flow, and the bulb does not glow. So, both these results indicate that air and distilled water are bad conductors of electricity. But when we short the two electrodes with this small piece of wire, we see that the bulb glows. That is because this wire is made up of metal, which is a good conductor of electricity. But what is this acid made up of, which on adding to distilled water completes the electric circuit and the bulb glows? It may not be as bright as in the case of the when we joined a piece of wire, but all the same we can see that the bulb glows. So, what made the acid to make the bulb glow? Now, this let us go step by step and see what is actually happening. So, first we had just the distilled water before we added acid to this. This distilled water is made up of water molecules, which is H2O. So, this is a model of water molecule. So, 2 atoms of hydrogen, 1 atom of oxygen, they form a covalent bond, and this is a covalent compound. So, these covalent compounds most of the times are not good conductors of electricity. Now, to this we added hydrochloric acid. This is hydrochloric acid molecule. Again, white is hydrogen and green is chlorine. The moment you add this to this beaker of water, distilled water, what happens is that the bond between chlorine and hydrogen are broken and hydrogen becomes free, and this free hydrogen can now go and bond temporarily with another water molecule like that, and this forms what is known as H3O plus. So, 3 atoms of hydrogen, 1 atom of oxygen, they form an ion called hydronium ion or H3O plus. Now, this ion is a carrier of electric current. Therefore, when you just have distilled water, there are no ions, therefore, there are no electric charge carriers, and therefore, there is no current flowing. The moment you put acid, acids form these hydronium ions, which form which are basically electrical charge carriers, and they perform the same function as this piece of wire, which when shorted the electric circuit is completed. Therefore, 2 things here. One, addition of hydrogen forms ions which are charge carriers, and secondly, it reduces the electrical resistance of the whole solution, because of which the conductivity increases, and because of that the bulb glows. So, this experiment clearly demonstrates that acids are good conductors of electricity only because they can form ions when put in water or when they are in a liquid state. Now, this particular experiment is an extremely important experiment. It just not demonstrates that acids conduct electricity. It also demonstrates a very, very important method which is very helpful for us to classify acids as strong acids or weak acids. Like I told you earlier, while all acids show common properties, there are acids that show a small difference in behavior from one acid to another acid. Similarly, this particular property of electrical conductivity can also be used to distinguish among different kinds of acids and classify them as strong acids or weak acids. What exactly are these will be the points of discussion in my next section. So, in the previous experiment with electrical conductivity, we saw that as we added more and more acid, we were actually increasing the number of hydronium ions in the distilled water that we had taken, and these increased hydronium ion concentration was responsible for electrical conductivity. So, what this implies is that any acid which forms a large number of hydronium ions will reduce the electrical resistance of the solution, therefore the electrical conductivity will be higher. Therefore, this electrical conductivity can be a very good measure of expressing how good an acid is. When we say how good an acid is, what we mean is how easily it can form hydronium ions, how it can form hydronium ions in very large numbers. Now, we saw that the same experiment with electrical conductivity, when we repeated with acetic acid did not show as much conductivity as hydrochloric acid. So, while both of them conduct electricity, it is very clear that the conductivity is not the same. Or in the language of chemistry, we say that both of them are acids, but the acidic character of acetic acid and hydrochloric acid are different. Now, it is one thing to say that they are different. In science, it is always important if this difference can be expressed in terms of some measurable quantity. And if we can put a number to this measured quantity, it is even better. So, around 1909, a Danish biochemist by name Sørensen came up with a fantastic method of measuring the acidic nature of a given acid, and he also came up with a method in which one could assign some number to a particular acid, and that number would actually indicate whether this acid produces large number of hydronium ions in distilled water, or it forms very few. And accordingly, he could classify acids into strong acids and weak acids. So, strong acids are those where hydronium ions can be very easily formed in distilled water. Weak acids are those like acetic acid, where hydronium ions are formed but very small in number. Why this difference arises between different acids is another very important question, but we are not going to answer that in this module. It is beyond our scope. Now, having said that, Sørensen came up with this scale of expressing how acidic something is, how strong an acid is, by putting some numbers. So, for the strongest acid he assigned a number 0, and for the weakest acid he assigned a number which was very, very close to 7, something like 6.99. In between 0 and 6.99, all other acids would have different strengths. So, if you can somehow measure this number, it is possible to say whether the acid is a strong acid or a weak acid. But the question is, how do you measure this number? How do you put this number? Hydronium ions are first of all very small, and they cannot be individually counted like you count the number of grains of rice in a given sample. Even that is not easy anyway, but even if at least it is possible, even though it is hard, but measuring the number of hydronium ions is very, very difficult. Therefore, we use the result of this electrical conductivity experiment to assign these numbers. So, he used a scale called pH. So, pH 0 represents acid with extremely high strength, that means it can form large number of hydronium ions, and a pH number of 6.99 would mean that it would have very few hydronium ions, and distilled water has this value of pH value of 7. Now, how do we put these numbers? Just as a blue litmus made use of a collection of dyes that could show a change of color from blue to red, there are certain chemicals which react with acids, but they produce different colors for different strengths. So, if there is huge number of hydronium ions in a particular acid, then the color of the paper, the litmus paper, would be something, and if the number of hydronium ion is very small, then the color of the same paper would be something else. So, instead of having a binary state of a blue litmus turning red or not turning red, pH papers, I have a pH paper here with me, and this pH paper is capable of turning into different colors depending on the different strength of the acid. So, let me demonstrate this by taking some distilled water in in this boiling tube, okay, it just becomes moist and a little translucent, and that is it, there is no change of color. Now, what we do is the following: we add a small quantity of acid, hydrochloric acid, which is our favorite, okay. A small quantity of acid is added now to this. Let us see if this small quantity that we have added is good enough for us to observe any change in the pH. Okay, so there. If you observe carefully, it has turned slightly pinkish at the edge or purplish. Color is not sometimes a very good way to distinguish, because different people have different sensitivity, okay. So, it changes color to just about a little purplish. Now, I add more hydrochloric acid to this. So, which means the number of hydronium ions that are being produced in this boiling tube is more. So, now we want to check whether this produces a different color, okay. So, there it goes. So, you find that it turns pinkish, which is very, very dark. Now, compare this with the previous pH paper that I had, which is much lighter than this. So, now if I were to dip this in hydrochloric acid, which is not mixed with much distilled water, let us see what the color change is going to be. Okay. So, there it is, which is not very much different from the previous one, except that it is slightly darker. Now, this is acetic acid. So, I take this into the boiling tube. Observe that it hardly changes it's very, very light pink, okay. Now, how do we find out the pH number from this? The pH number from this can be read out. So, now you try to compare the color code that is there on the pH paper with the color that the pH paper has turned to, and that will actually indicate what the pH number of this particular acid is. So, Sorensen came up with this kind of a measuring method. So, by assigning different values of the pH number to the acid, we can see whether the acid is very strong acid or whether it is a weak acid. So, 0 for strongest and 7, nearly 7 for the weakest one, okay. So, we are still using some kind of an indicator. So, while the blue litmus indicates whether the acid is present or not in some mixture, pH paper actually tells you whether the acid is strong or not. So, again it is an indicator based reaction. In addition to pH papers, you also get these universal indicators, okay, like universal indicators. So, when you add this universal indicator to a given acid, the acid changes its color, okay. And again you will find there is a color code on this universal indicator. So, depending on the color that you match, you can actually find out what the pH value is, okay. So, just to see the effect of adding water, so what we can do is the following. Okay. This is distilled water, and we add only a small quantity of this acid and stir it. So, we have small quantity of acid added to distilled water, and now we see what happens when this indicator is universal indicator is added to it. So, it still shows a good pink color, which is pretty close to a pH value of let me read out from the bottle. It gives you something like a pH value of 4. So, pH value of 4 is a pretty strong acid. It is not weak by any measure. So, which means even a small quantity of these hydrochloric acid in a large amount of distilled water can still be very, very strong. Therefore, it is not surprising that hydrochloric acid is classified as a strong acid. Now, to summarize this particular part, what we have seen is that using pH number, it is possible to classify the acids as strong acids or weak acids, and pH number itself is obtained either by using a universal indicator or by using a pH paper on which you have a color code. And the basis of making this pH scale is again the electrical conductivity. So, depending on how much of conductivity is shown by a certain acid, it is possible to calibrate a scale and give pH numbers that we see here. And in the meanwhile, while I am preparing for the next module on this, you can go over this. If you have questions, you can get back to us. The email ID is given at the end of this particular video. So, thank you very much.